Now, as we continue looking at sex determination, sex linkage, sex limited, we start looking at all of those type of problems. Let's start off with, again, males are XY, females are XX. We have just a basic Punnett square showing you a female egg that's XX, the male sperm that's XY. If we look at this, the male is the one that determines the sex of the child. If he gives an X, they have a daughter. If he gives a Y, they have a son. Now remember that the Y chromosome carries about 231 genes. So genes on the Y chromosome are said to be Y-linked. These are very rare. They can only be passed from male to male because, again, the father, if he gives an X, it's a female. If he gives a Y, it's a male. So no affected results on females. Currently, most of your Y-linked traits are going to be involved in some kind of infertility, or they're not transmitted at all and they don't show up. X-linked traits are carried on the X chromosome, so that a male, the way we write this down is we would put that trait right there on that X chromosome, and that shows that it's an X-linked trait. Now, because a male is XY, if they get an X chromosome, and they would get it from their mother that carries that recessive condition, then they're going to exhibit that condition. For a female to have it, she has to have the trait on both X chromosomes. So she has to get it from both mom and dad. If she gets the trait on only one X chromosome, and it doesn't matter which way we draw that, but if she's got it on one of those, then she's a carrier. She can pass it on to her children, but she will not display the trait. So X-linked recessive traits are carried on the X chromosome. You need to, when you do the problems and for your test, you need to indicate it on the X chromosome or it will be considered wrong. If a male gets the X chromosome with the condition, he has the condition. For a female, she has to get it on both chromosomes to exhibit the condition. Otherwise, she's just a carrier. Some examples of some X-linked recessive conditions: hemophilia, which is the inability to have blood clotting; color blindness; ichthyosis. Now, ichthyosis is what we would call the alligator individual. This is a deficiency of an enzyme that removes cholesterol from the skin. The skin becomes very dried and cracked. So here you have a picture of what ichthyosis looks like, and then you can just see just a pedigree of how it's covered, being passed from the mother down to the son. The son gets the X chromosome, they're going to have the condition. The daughter, now the daughter's a carrier, but the only way the daughter would have the condition would be if the mother here had the condition as well. This is the pedigree of the Romanoff family. So if you've heard of Anastasia, this is her family tree. The hemophilia trait was passed down from Queen Victoria, and it passed through the family. Ultimately, in Tsar Nicholas of Russia's family, his son Alexis received the trait. That is how history was affected, with Rasputin becoming a close confidant of the family, trying to save Alexis. He betrayed the Romanovs, and then they were overthrown, and eventually they were killed. Now, X-linked dominant inheritance is a little bit more rare. So on this one, if the male receives a trait, they're going to get it just like before. So there you go. But on the female, because it is dominant, she only has to receive the trait from one parent, and then she would have the condition. So this is going to be much more severe in males. A lot of miscarriages in males before the babies are born is going to be due to an X-linked dominant inheritance. It can be passed from males to their daughters, but it cannot be passed from a male to the son because he gives the Y chromosome. So an example of an X-linked dominant trait is incontinentia pigmenta, and on this one right here, the newborn baby that has this condition, and they shorten it to IP. They have yellow pus-filled vesicles on their body. The lesions will rupture, they become warty, and then eventually the warty portion goes away and it leaves brown spotches all over the body from where they were affected. And so you can see the diagram over here. You can see some of those brown splotches. Another X-linked dominant trait is hypertrichosis. Hypertrichosis is the condition that gave rise to the belief of the werewolf. You will actually be doing a lab later on this unit dealing with hypertrichosis. I do have a separate video that shows you how to approach solving the genetic problems. I do have the steps here, but I'm going to refer you as you're doing your homework. Watch that video because it will show you step by step through different examples how to approach to solve the genetic problems on your upcoming assignments. A sex-limited trait is a trait that affects a structure or a function in only one sex. For example, beard growth, or milk production in females, or preeclampsia in pregnancy. These traits can be autosomal or they can be X-linked, but again, the key is that they show up in only one sex. This is an example of a sex-limited characteristic. It deals with hen feathers, and what I would like for you to do is make sure you pause and you copy this key down because you will need this key on some homework problems, either on the sex-linked or the multiple allele assignment, so make sure you mark this. A sex-influence trait is a trait in where the phenotypic expression varies on the heterozygote. In other words, male pattern baldness is an example. So if we look at male pattern baldness, and here we have the key for it. Well, big B would be bald in both men and women. Little b, both men and women would not be bald. It's that heterozygous right there. So if they get the big B in men, they're going to be bald. In women, they would not. So the big B is going to result in baldness in men regardless. In females, for them to be bald, they have to receive the big B from both parents. Make sure you write down this key as well. So males are XY and females are XX, meaning females have 2 X chromosomes, but males only have 1. So what we have occurring is X inactivation, and this is going to balance out the fact that males have 1X and females have 2. And so in females, one of the X chromosomes will randomly be inactivated in each cell. And this inactivated X chromosome is called a Barr body. If you do a cheek cell, you can look at a cheek smear and look at the cheek cell and determine just from the cheek cell if an individual is a male or a female by looking for these Barr bodies. The X inactivation occurs early in prenatal development before the individual is born. It is an example of an epigenetic change, meaning the inheritance may change, but it's not because a change in the DNA base sequence or the ATGCs has occurred. So you can see these Barr bodies right there, and those Barr bodies are found in females, but they're not found in males. So this was discovered in the 1940s, and it was discovered in female cats first. And today we know that you can look at cheek cells, look for the Barr bodies. If they're present, it's a female, and if they're not, it's a male. Now, a good example of X inactivation is in calico cats. We'll show you the key on the next slide. So the key for calico cats, if it's a male, what we have going on is right here. This would be a brown male. Here we would have the yellow male. Here we would have a brown female, a yellow female, but to be calico, they have to have a trait for the brown and a trait for the yellow color. Now, occasionally you'll hear people talk about, oh, they have a calico male. Well, for a calico male to exist, it actually has to be a genetic abnormality, and they have to be XXY, and that's called Klinefelter's male. The male would be infertile, and what happens is they have two X chromosomes, an X for the brown color, an X for the yellow color, and then they have the Y. So theoretically, the X inactivation is supposed to even out the expression of the X-linked traits between males and females. It doesn't really do that. If you think of the number of individuals who are colorblind, you're always going to come up with 5 or 6 that are males, and maybe, and all the times I've taught, I've never had a student say, oh, I know a colorblind female. And the reason the females have 2 X chromosomes and the males only have 1. Let's look at another example of X inactivation. A situation where the heterozygous for the X-linked trait can be extremely harmful is the craniofrontonasal syndrome. And so an individual, which would be a female, that is heterozygous have very severe phenotypic expressions. They have a very abnormal face. They'll have abnormally fused skull bones. And then this little girl right over here is an example of an individual that is suffering from this condition. Males and females that are carriers for this condition, or where that one X chromosome is turned on, they'll have asymmetrical face features, but nothing like a heterozygous female. Now, a condition that just nearly always affects females only is called Rett Syndrome. The symptoms for this condition is where the girls do strange things with their hands. So they may have an uncontrollable tapping of objects. They may be clapping their hands or putting their hands in their mouth. Or if you look at the picture of the girl over here, they may have a condition where they wring their hands a great deal. It is dominant, and in 99% of the cases, it is not passed from parent to child, but it arises brand new in that child. So genomic imprinting, when we look at this, the phenotype of an individual is going to differ depending on what genetic traits they received from their parent. Genes in genomic imprinting are imprinted by an epigenetic event that's called DNA methylation. And methyl, which is just CH3, it binds to the DNA. It will suppress, had trouble saying the word, it will suppress the gene that expresses a specific pattern, and that is determined by the individual's sex on whether the pattern will be expressed or not. So that the imprints are erased during meiosis, and then they are reinstated according to the sex of the individual once fertilization has occurred. So the exact importance of genomic imprinting is still being studied. The functions aren't really well understood. They believe it might play a role in some developmental process. Research is saying that it takes 2 opposite sex parents to produce a healthy embryo, and the genomic imprinting may also explain incomplete penetrance of a trait not completely showing up in an individual. So there have been 2 distinct syndromes that have been identified relating to imprinting, and it deals with the deletion in the chromosome 15. And the syndrome is based on whether you receive the trait from dad or from mom. If you receive the chromosome with the deletion from dad, then the child will have Prader-Willis syndrome. If the child receives the chromosome with the deletion from mom, they will have Angelman's syndrome. So there's two different syndromes that are a result of a deletion in chromosome 15, but it depends on whether you receive the inherited gene from dad or from mom. And so this one right here is Prader-Willis syndrome, and this is when you receive it from dad. And this right here is an individual that has Angelman's syndrome, and they receive that particular chromosome from mom. With Prader-Willis syndrome, this one is due to the loss of the function of specific genes that are missing on the chromosome 15. So in newborns, the symptoms would include weak muscles, poor feeding, and slow development. As the individual grows, when they reach childhood, the person becomes constantly hungry. This can lead to obesity, type 2 diabetes, and mild to moderate intellectual impairment. They can also have behavior problems as well. The physical characteristics of Prader-Willis would include a narrow forehead, small hands and feet, short height, and light skin and hair. With Angelman syndrome, it mainly affects the nervous system, so the individual may have a small head, they'll have severe intellectual disabilities, they may have speaking problems, and problems with balance and movement. They may have seizures, they may have sleep problems. These individuals usually have a happy personality, but as one becomes older, especially by the age of 1, you're going to start to notice some of these symptoms showing up more and more. As far as the physical appearance, small head, wide mouth, very happy demeanor. The hands can have a tapered finger appearance, have abnormal creases and broad thumbs.